Hello everybody and welcome to today's webinar. On behalf of European Pharmaceutical Review and Rapid Microbiosystems, I'd like to thank everyone for attending. My name's Caroline Peachy. I'm the editor of European Pharmaceutical Review and I'll be your host today. Joining me today is Dr. Tim Sandel. Tim Sandel is an independent microbiology consultant. He's got over 25 years of experience in microbiological research, biopharmaceutical processing. He's the author of more than 30 books on microbiology, and he's a visiting tutor at both the University of Manchester and UCL. Tim's here to present on the advantages of automated microbial colony counting. Welcome, Tim.
Okay, thank you very much.
We're also joined today by Dr. David Jones, who's a Director of Industry, Regulatory and Government Affairs at Rapid Microbiosystems. David, too, has more than 20 years of experience in rapid microbial detection technology and development of lab instrumentation. Welcome, David. But now, without further ado, I'd like to hand over to Tim to start our webinar today.
Okay, so this webinar is entitled The Advantages of Automated Colony Counting. And in the webinar, we're going to be covering a number of issues to do with colony counting in general, and then we'll gravitate towards automated systems and some of the advantages that entails. So we begin with looking at the colony forming unit and what it means and how we count and some of the concerns that arise, particularly with the limitations of human visual acuity and some of the complications that can emerge with the way certain colonies grow on plates. And we'll also have a look at some regulatory trends and some regulatory citations that have arisen which describe some of the limitations and some of the problems that inspectors come across. And they also then lead us into the automated technologies, and we'll have a look at how some different systems work and some of the advantages of synergistic systems and some of the validation questions as well. So let's begin with just an overview. If applied properly, automated colony counters can provide faster and a more reproducible means in order to capture and interpret microbial test results. And a key practical advantage with automatic colony counting is the ability to transfer data from what is seen directly into a computerized system, which offers some strong data security factors. Central to the validation, microbiologists need to be certain that the automated colony counting technology is indeed as accurate or superior to the manual counting method. And also that any data is secure and that it's free from manipulation. And with all this in place, then the accuracy of the counting can be improved and data security can be enhanced. But to get to this position, we need to work through the process of colony formation and the more traditional routes for colony counting. So with microbial colonies, I'm sure most people online will be aware, but a colony is a representation of a viable cell, a cell that is able to grow on a growth medium, typically a standard agar plate, and to form a population. Now, the growth of a single bacterium in a defined location in the agar plate can give rise to this colony that eventually becomes visible over a period of time, and depending upon the organism, the incubation conditions, the atmospheric conditions, and the formulation of the agar, that might take 1 to 3 days, or it could extend to a much longer period of time. And there's always going to be eventual restrictions on the colony size. That's affected by viscous drag and other forces that are exerted, particularly by neighbouring cells. So these will restrict the size of the colony that can form, and there's always going to be competition for space if we have a higher number of organisms that we're trying to grow. But the important thing about a colony is the actual word colony, which connects to clonal. With all of the organisms, the organism that forms the colony, if it's not mixed, then it's theoretically descending from a single ancestor, and all of the cells will be genetically identical, except for any mutations that might occur. Mutations will always start at a relatively low frequency. So that's colony in general. But then we have this concept of the colony forming unit. So the colony forming unit utilizes a microorganism's capability to replicate on an applied medium under particular temperature and time conditions. But the CFU is always going to be an arbitrary estimate because, first of all, only those cells that are able to form colonies that can grow under the particular conditions of the test will produce a visible colony, and this is going to be dependent upon incubation temperature, incubation time, the type of culture media, the oxygen conditions, and so on. And also we can have more than one organism that's come together that appears to form one colony. All we get is the representation of that colony. We also have different ways to describe organisms in different colonial states, and we may have typical and we may have atypical growth patterns. And there are some descriptors that we can use to capture that. So we can talk about form, by which we mean the basic shape of the colony, for example, whether it's circular or filamentous. We also have the size, the diameter of the colony. So we might have very tiny colonies, which are sometimes referred to as punctiform, and then larger colonies. We're also going to have the elevation of the colony, which is the side view of the colony, the margin or border, which is the edge of the colony, which is often clearer through magnification. Then we have the surface of the colony, whether it's smooth, glistening, rough, wrinkled, or dull. And then we have the opacity of the colony, whether it's transparent or opaque or translucent. And then there is the colour, the pigmentation of the colony. Some colonies are white, some are buff, some are red, and so on. So from one point of view, colony counting might appear straightforward, but there's a lot of evidence to show that errors can happen, and this can either be through failing to spot colonies due to their appearance, their size, the way they're very close to each other, their colour that makes them difficult to count, or through the issue of operator fatigue. And that can be partly dependent on the sheer number of plates that are there to be counted. There are also problems that arise in general. On the slide now, we have a photograph of problems with colonies that are either merging or clumping together. So the plate on the left-hand side is a bioburden sample, a sample that's been plated, and there's a bacillus-like organism. You've got a lot of clumping, the sample's not been adequately mixed. And then we have on the right-hand side, we've got two colonies merged together, or maybe it's more than two colonies merged together. It'd be something that would need to be looked at a little bit more closely. On this image, we have a problem of poor mixing, again with a pour plate, and samples being pipetted in, the plate's not been adequately mixed, and we've got a congregation of colonies on one side of the plate, and starting to get some confluence there that can make it hard to discern the actual numbers. And here we have a settle plate, and this is a cracked plate, so it's based on desiccation, and this may have arisen from a settle plate that's exposed within a unidirectional airflow, which is a dry and relatively harsh environment. So perhaps the agar fill wasn't sufficient, or the agar gel strength wasn't robust enough. So we have this kind of cracking and loss of moisture. Now, a regulator would regard that as a data integrity issue because you've got a portion of the plate which could have contained a colony which is no longer visible. Here we have what is some apparent growth off agar, which is rare, but again, can sometimes occur if there's a thin layer of agar on the edge of the plate. So that can cause problems as well. So there are issues with actual microorganisms and the way they grow. But equally, the process of colony counting is open to different elements of people, what you might call human error, although anyone familiar with root cause analysis will know that there's the requirement to go further and to get to the true causes of human error. But problems can emerge in terms of counting and also with transcribing data onto a computer. Some of these things can be factors like eye strain, or with simply transcribing the wrong result. And these issues can be compounded where we get more atypical microbial growth upon agar. And we also will have colonies that look different. So the growth on an agar plate, particularly with a mixed population, is not always going to be homogeneous. So we're going to get colonies with different diameters, densities, shapes. Some will show confluence, some may merge, some may spread, some may clump. And all these sort of clustering issues and things that we've looked at in the previous images. There's also going to be issues about what we can actually see. So we're into the kind of concept of colonies that are there but not always visible to the naked eye, or they might be more visible to one person than another person, or there might be factors of reading that plate that affect that process. So there's always going to be differences in the way that our eyes and brain interpret the perceptions of colour, shape, depth, movement, and other features. And the ability to do this is going to vary among different people. It's also something that alters with age. So for these reasons, where we are carrying out more conventional colony counting, again, as shown on the picture, there's a standard colony counting device, then assessing vision periodically for someone whose job is to count colonies in the conventional sense is quite important to make sure that person is still able to count colonies over time. And an important assessment, an important visual assessment, is to the level that might be undertaken for those who are tasked with inspecting pharmaceutical products, and that comes down to the concept of visual acuity. And this refers to our ability to discern the shapes and details of the things that we see. So it's about being able to distinguish fine detail, and it's one of the factors that constitutes our overall vision. And to this we can also add colour vision, peripheral vision, and depth perception, and all these combine to create how we see and how we interpret the world. There are also other factors that influence counting and which can be introduced to improve the counting of colonies. Standardizing viewing distance is important. So again, the concept of vision shows that there will always be an approximate distance between the person looking at the plate and the plate they are reading. That's going to vary a little bit between different people, but a standardized assessment suggests that's around 400 millimeters. There's also the need to have appropriate lighting conditions, and we can express that in either lumens or lux, but there's going to need to be a bright enough light source, and again, the use of a white light is generally preferable to an amber coloured light, or the type of light that might be more conventional in, say, an angle poise lamp. And that's again a factor where, particularly where we're looking for sort of buff coloured or beige coloured colonies on a medium that is already that similar colour, a white light source gives greater clarity than an amber light source, for example. The viewing angle is important, and also to be able to have a magnifying glass as well for a scan after the initial count to make sure there aren't any smaller colonies that are necessarily visible to the naked eye. Now, also, you may be wondering, we've been counting colonies for years and nothing terribly has ever gone wrong, but errors can and do inevitably occur. So there was one study that was published, and this set out to examine the false positive rate, and it was this. By this, it means the overcounting of colonies, and it also set out to examine the false negative rate, which is the undercounting of colonies. So to assess this, around 1,000 plates were read by different operators, and these operators were based in 3 different laboratories. So that combination of number of operators, number of laboratories, the 1,000 plates led to about 13,000 plates being read in total. And the plates were designed so that 79% were negative and 21% were positive. And this was done by the use of tiny beads to represent colonies. Now, these beads ranged in size from microns, and some of the beads were coloured black and some were coloured white. So there's a number of design elements that were really there to challenge the operators as much as possible. And the number of beads per agar plate was either none, obviously for the plates that are designed to be negative, and then for those that were positive, they ranged from 1 to 12. So they're trying to represent lower levels of contamination, the sort of levels that could be in grade A, B, or C, clean rooms, for example. And the results were very interesting. They showed a variation between operators, and this variation between operators was to the extent that some operators could misread the positive plates by up to 50%. The negative plates were generally read satisfactorily, so there weren't really any cases of people seeing colonies that weren't there. The bigger risk factor was with undercounting colonies that were actually there. It also stands that the results across the 3 laboratories were generally very similar, so the same error rates were reproducible. The error rates were far more common with smaller colonies than with larger colonies. But actually, the biggest influence for correct counting or incorrect counting was the position of the colony on the plate. It also stands that counting errors were greater for the lighter colonies, so it's those that were represented by the white beads, than it was for the darker colonies as represented by the black beads. So this also led to some inferences, and these inferences were that colonies that are between 53 and 63 microns, really the chance of an operator seeing these and counting these was close to non-existent. Colonies between 90 to 166 microns can be counted, ideally under conditions where there's a strong contrast between the colonies and the background, a darker field for lighter colonies and a lighter field for darker colonies. But even under these more ideal counting conditions, errors in counting were still relatively common. For the counting of colony sizes between 212 and 250 microns, then the data was better. but the position within the agar plate was still an important variable that can impact upon counting accuracy. The greatest counting accuracy, the most successful outcomes, were for those colonies in size from 425 to 500 microns. And for colonies of this size, then the position in the agar or the fact that they were white or black coloured didn't make any difference. So this was a sort of an optimal range. So the final review placed the greatest level of error on simple limitations of the human eye or the process of visual acuity. So it wasn't because operators were being distracted or they'd read too many plates. It was simply that it is a challenging activity to do. And this led to a conclusion that such errors were more likely to be overcome with automated systems. And some of these issues that I've mentioned are borne out through regulatory concerns. So issues with plate counting have been raised by European inspectors, MHRA, and they also appear periodically in US FDA warning letters. And we get greater clarity of that because of the FDA openly publishing findings more than agencies. But some of the things that kind of appear in these sort of FDA concerns include failure to verify counts between operators, failure to use suitable equipment to assess colonies, so not using appropriate light sources, not using colony counters at all, not using magnifying instruments. There's also been flagged differences at the counting that occur between operators, counts that are evidently on plates not matching what's been recorded down on a computer system or on paperwork, or loss or damage to plates before the data can be captured. And within the FDA question and answers on data integrity, there's a indication that manually counted plates should be verified by a second person. So this is a tendency that doesn't appear to be as strong amongst European inspectors, but it is a growing undercurrent within US inspectors. And there are also things that arise from laboratory bits and inspections and questions of supervision. So clustered around the way that data is processed and reviewed, what controls there are around the potential for data manipulation and deletion, the criteria that could be used to invalidate data, how data is transferred onto systems, whether that's something that's written down that then typed into a computer or recorded onto a report form or through some other means, and how that data is checked and verified and shown not to alter in its value or in its meaning. If there is any degree of second checking or how results are reviewed is what that's based upon, and whether that's statistically sound for computerized systems, what the level of access controls are, and other supporting factors like electronic signatures, how data is archived, and also the appropriateness of metadata as well. So what time was the sample taken? What's the unit of measurement? All these kind of supporting data is equally as important. So returning to the regulatory piece, I just pulled out some recent FDA warning letters, and just to paraphrase the first one, obviously the FDA inspector found some colony forming unit values that were higher than what was necessarily higher on plates of necessarily what recorded in the actual records. So this led to the declaration that there'd been falsification of data, whether there was or whether it was purely lapses or errors or there was something deliberately going on. There's also some other examples where CFU are miscounted or there's missing samples or samples that have been dropped and then written off because, oh yes, I know there were so many colonies on the plate, but the lid was locked or something. So it always gets suspicion in the mind of regulators, and it's not been unknown for inspectors sometimes to recount plates or even to take plates out of waste bins and re-examine them. And that isn't an urban myth. That's something I've actually seen happen myself. So there are some clear advantages to consider automated counters, automated colony counters, and there are different technologies that are available on the market. So we need to consider what makes for a good system. And the most suitable automated colony counters are those that will be unaffected by the inoculation method, by the shape or size of the colony. And an effective automated colony counter will need to have good sensitivity for the detection of smaller colonies, particularly in low contrast media. We'll also need to have the ability to capture counts and images using digital technology, so we have some evidence of what was counted, and then have the ability to transfer the data, such as to a laboratory information management system. And more recent advances as well have the ability with systems that can also learn as they go along, taking out the advantages of machine learning as well. Now, central to the automated system is having a good optical response, because the optics are essential for differentiating what is a colony and what isn't a colony. So there needs to be good optical response performance. So this is having sufficient resolution and control of things like background noise, contrast, and resolution. And this is all achieved through camera and image acquisition tools. So we'll be wanting effective image resolution. And this will mean we need to be able to get files of sufficient sizes and have good data management. And we want to have also, for the resolution, is good sample lighting, and we want a uniformity through the series of images that would be. We also want the ability to visualize all different sorts of colonies of different formations and different colors. So this might include chromatic and achromatic images, and be able to deal then with clearer media and more densely colored media. We want the system to be able to separate out aggregated colonies. We may want the option to count an entire plate or potentially sectors of a plate, depending upon if we're just doing, say, certain serial dilution schemes, for example. We also want relatively fast throughput, and certainly we don't want an automated method that taking far longer than a conventional counting method. And for looking at certain plates and wanting to show certain people, we'd want the ability to display on-screen images or have ability to capture photographs for later viewing. Now, not all automated plate counting technologies are identical, and some are less successful with dealing with mixed cultures or the growth effects that might lead to merged colonies. So we can get problems of underestimating the numbers of colony forming units on a plate, and that might occur to the presence of other debris or due to plate overcrowding. Another problem that can occur is the failure to discriminate colony overlap, which can occur through things like masking, together with concerns about interpreting the degree of convexity, colour brightness, variations in colony shape, differences with colony texture and so on. There can also be, and if you hark back to one of the images we looked at earlier, a failure to assess colonies located near to the plate periphery, and also with failing to accurately count colonies where increased density occurs, and also if we've got variations in pigmentation where we've got two very differently coloured colonies very close to each other, that can sometimes cause problems, false positives as well. So this could occur if the system is unable to adequately differentiate between colonies and other debris, or with the formation of any air bubbles that might occur inside agar, and that can be more common in plates that we might be pouring ourselves within a laboratory. It can also sometimes occur with pre-purchased media as well. It also stands that counting procedures can also lead to deformities in the agar as well. Artifacts like imperfections in the agar, dust, a lack of clarity, particularly with certain media that we might need to add an additional ingredient in during the preparation process, and we have to then sterilize if we sterilize the media at a lower temperature, and sometimes they can lead to kind of swirl-like patterns within the media itself, which can then cause issues. There are also other influencing factors that can affect the upper countable range, and these can include colony size and behavior, possible swarming, which can occur with certain gram-negative organisms in particular, so I think organisms like Proteus mirabilis, for example. And we can also get clumping effects, and this is a particular concern with a number of species of Bacillus, and Bacillus cereus is particularly for this kind of desire for cells to gravitate and then colonies to gravitate towards each other. In terms of the other end of the scale as well, we want a limit of detection and a limit of quantification that go down to a single colony. If we're a sterile products manufacturer and we're processing in grade A, then it's crucial that we can assess that difference between 0 or 1 colony, and sometimes that can be the difference between batch acceptance and batch rejection. So the kind of key features, if we're beginning to draw up our sort of imaginary user requirement specification for an automated system, is that we want standardized and accurate results. And accuracy is important because of these various things that can affect that, the various things that we've mentioned. We also want an ability to count colonies within appropriate parameters down to a certain level, say 50 microns, and to measure zones accurately as well in terms of the differential space between colonies. We want this good optical response, its ability to deal with background noise, contrast, and resolution. And we also want to be able to collect files of suitable sizes. And we also want to be able to deal with this issue of aggregation and to be able to also zoom in if we need to look for smaller colonies. So this leads to achieve this, there's going to be a series of technical requirements that we'd also need to consider. So we'd want to be looking for things like a circular dark field illuminator. We'd want to have a camera with good resolution, and generally the sort of minimum is around 3.3 megapixels, and often we want to go for cameras with higher quality. The software and the appropriate algorithms that drive that software need to be suitable for interpretation, and if we can have something that even learns over time, and if we do need to make the occasional correction, that then becomes inbuilt into the system going forwards. We also need to consider the way that plates are held and the degree of automation that exists with that, and the degree that the plate is held in a way that's stable and not prone to vibration, because again that's important for both the image analyzer and the software. And also we need to understand how the algorithm works, how it chooses to classify what it sees, how it assesses the geometric properties of what it's looking at, how it looks at things like major and minor axes of the colony, and how it really defines what is and what is not a colony. Another important technical factor is the illumination and detection system. And these are going to vary according to different types of automated counters. So some might be full spectrum white light bulbs, other will use narrow wavelength these, and some will use very narrow wavelength lasers. There's also differences in the way that data is captured. So this could be a photon multiplier tube, for instance, or it could be a charge coupled detector with some form of wavelength selecting filters. Again, there's going to be differences in outcome that can arise from that as well. And some counters can be optimized for use with ultraviolet light illumination, some with white light, some with fluorescent. Some might need to be set differently for the colonies that produce fluorescing pigments. And again, that can be a more challenging function in the case of the pigment produced by Pseudomonas aeruginosa, for example. So there are other sort of issues to consider. There are also some automated systems that have the advantage of being able to detect colonies early, so looking for very tiny colonies that would not be visible to the human eye, looking for some micro colonies, and these require advanced optics, scientific grade cameras, and there are technologies that can go down to 43 microns, which is we looked at before, is on the level or beyond the ability of the human eye. And a number of these technologies can use factors like blue light excitation to detect and to kind of capture and indicate the presence of these micro colonies. It's also important not to see the automated colony counter as this kind of standalone system. Yes, you can feed plates in and you can produce results relatively quickly, but there are also advantages that arise from synergistic systems as well. And this enables us to develop more meaningful and lean and efficient workflows, to reduce errors through things like barcode functionality to improve the traceability of samples. This also then helps to reduce instances of losing plates, and also to reduce instances of if we've made an error somewhere else, like incubation error or something like that, or if we can build the incubator as part of the automated counting system, then we can use robotics to avoid those errors completely, and also avoid errors if we wish to undertake something like dual incubation, which is a very common step within environmental monitoring practices. And we can also avoid miscounts, reduce transcription errors, avoid the human introduction of contaminants, and also preserve the integrity of plates by less chance of dropping them or leaving them in incubators for too long. One way to promote these better synergies is through the use of robotics. And here we can increase capacity. We can use systems with multiple robotic arms to move plates to different incubation stages. We can have automatic loading systems to accommodate an entire system. And if you think about monitoring, aseptic monitoring, I've used an example on the slide, is often where you can generate the most number of plates from a single session, and you can capture all of those within one go through one system and one solution. Systems that are able to work on the basis of continuous processing through their incubation prior to reading often have the flexibility of being able to have additional plates added, and then the eventual counting and capture of the data. And that capture of the data obviously leads on to some important data integrity considerations, and any automated robotic system is going to have to meet the number of the key data integrity requirements that apply to the laboratory. And there are a host of questions, and 5 which come to mind are: is the electronic data available? Can it be accessed from wherever it's been archived? Is the electronic data reviewed, and what levels of controls and passwords are around that? Are audit trails reviewed regularly? Is the metadata assessed? Is there clear segregation between the person responsible for operating the instrument with a separate independent administrator? And how has the system been validated for its attended use? There are other important considerations as well, like access to clocks, user access rights we discussed, how things are printed, what happens to the printouts in terms of, and what's considered to be the primary source of data. Validation is also of great importance, and the microbiologists leading this activity would need to establish how they were going to set out to qualify the automated counting and to come up with a way that is as precise or superior to the manual method, and how that fits into the workflow. So for validation minima, this needs to be considered across the laboratory's maximum and minimum countable ranges. It will need to include both pure and mixed cultures. It would need to include multiple assessments of the same plates as well as a range of plates. There needs to be an acceptable range of error, whether it's plus or minus 1 colony or no acceptance, or whatever the criteria for undercount or overcount is. And that needs also to be assessed in terms of a suitable confidence interval and a statistical test for significance. And there are different ways to approach this. The simplest way is to recount using the conventional manual method, using a pen, and have different operators to verify what the automated system is producing, to ensure that a range of different types of organisms, different agars, different colony ranges are used, and in particularly plates with mixed cultures, and very different looking colonies as well, as we discussed at the beginning: smooth, shiny, bright, opaque, low, high convexities, and so on. So those real robust challenges are quite important. And to begin with, it probably makes sense to use pure cultures of known organisms, because if the system can't count those, then there's little point going forwards, and then move on to more robust challenges, looking at variations in counting ranges, a mixture of environmental monitoring, bioburden, streak plates, raw materials, in process materials, and so on. So really challenging those kind of real life scenarios, because otherwise validation can sometimes go wrong. And some of the errors are failure to differentiate mixed colonies, not able to do with the sufficient space between colonies, variations in agar thickness, so it's not possible to discriminate all areas of the plate, weaknesses where there's confluent growth, inadequate light, and whether that works better with white light or blue dark field illumination, and there's going to be variations with technologies, and how the presence of debris is also looked at. And then, as we've mentioned, data verification is also important. So we want to avoid any ability to manipulate the data. We want to be able to archive, retrieve data, transmit data to other computerized systems. We want to ensure that we can give everything a unique identifier, and that we have the ability to take and retrieve photographic images of what we've been looking at. So this completes the main part of the webinar. Just in a nutshell, for the objectives of plate counting, we're seeking greater accuracy of counts, we're seeking improved data security, we're seeking improved throughput, and the automated solutions are a far more robust and reproducible way